We've got a fantastic group of speakers lined up, and we're going to get going with the session now. As in previous sessions, if questions could go into Slack, that would be ideal, although we will also monitor the Q&A function on Zoom for questions at the end of the talk. So the theme of the session is looking at structures and refinement using multiple methods and multiple approaches. Our first speaker is Dr. Dorothee Liebschner, who is a bioinformatician from Berkeley Lab within the Phoenix project. And she's been quite widely involved in methods development for macromolecular crystallography and in combining data obtained using electrons, neutrons, and X-rays. And today she's going to talk about a range of topics, including differences in maps obtained using these different methods and how this can be linked to validation approaches. So if you could go ahead and share your screen, I will hand over to you.
Okay, so I'm sharing my screen. First of all, can you confirm if you can hear me, if the mic works correctly? Okay, I cannot hear you now, but okay, and now I will go on to presenter mode. Does this work correctly as well? Okay. And right, I will stop my video because sometimes I do have a spotty internet, and without video it works just better. And if there's anything with the connection, please shout loud, and then I can stop and maybe restart where we were. Okay, so my talk is about structure determination using data from different sources, X-rays, neutrons, and electrons. And so let's revise, first of all, the different methods for structure determination. So first of all, it's diffraction or crystallography. For this kind of experiment, we have a crystalline sample that we will illuminate with some radiation that can be X-rays, neutrons, or electrons. Then we will collect the diffracted beams on a diffraction pattern, and the reduced data is a set of intensities as a function of HKL. For cryo-EM, it's a little bit different. Here we have an electron beam that is shot on the sample, which is here on the grid. Then again, we collect some images, and when we process these data, we can obtain a 3D reconstruction, which is basically a 3D map. So for any of these methods, what we're doing is often we analyze Fourier images of maps. So here's an example of that. It's an example of an electron density map. And it's sufficiently high resolution, these density peaks correspond to atomic positions. So we can build a model into that map. So here we have now a model overlaid to that density. So while this is generally true for all three methods, there are subtle differences between density maps from different methods. So here I have an aspartic acid residue, and I calculated a model-based density using for these three different types of interactions, so X-ray, neutron, and electron. And on purpose, I didn't actually put any label here because I want you to think about what actually each of these images represents. And if you don't know, what we're going to do for the remainder of this talk is we will discuss what are the differences in the maps and where do these differences come from. And so just to keep it simple, let's focus on crystallography, because then we can use the same formalism and we don't have to swap, let's say, names of variables and so on. So let's go back to our crystallographic experiment. So we have radiation that we shoot on our crystalline sample. And now the density rho of the matter in the unit cell is related to the amplitude and phase of the scattered radiation. So this is the formula most of you should be familiar with. The electron density is the Fourier transform of the structure factor. So here this quantity F is the structure factor. Now the structure factor can be itself expressed as a sum over all atoms of these little FJs here. And FJ is the so-called scattering factor. And now we see on the left-hand side, we have density, so that is related to a map or the Fourier image of a map. And on the right, we have the scattering factor. So if we want to understand the features of maps for different methods, then we have to understand what the scattering factors for these different methods look like. So what are these scattering factors? So the scattering factor, small f, is basically the Fourier transform of an atomic spatial density distribution. So it's in direct analogy to the structure factor capital F, which is the Fourier transform of the density in the unit cell. So I want to point out here the scattering factor is the related to the atomic density. Now here, I'm not saying what kind of density, because that depends on the type of interaction. Now, in the previous slide, I used the notation H for the scattering factor, but because the scattering factor is not only used in crystallography, often people use the notation S for the scattering factor. And then R is the coordinate in real space. So the scattering factor is the measure of the scattering amplitude of a wave by an isolated atom. And what we're going to do now is we compare for the different radiations, X-ray, neutron, and electron, several features of these scattering factors. So we will basically fill out this table for each of these three interaction types. So we will look at the scattering factor and we will think about what influence this will have on the map. And I want in particular to focus on neutral atoms, on ions. That means there's a charge or an excess or some electrons are missing. On isotopes, that means these atoms, the nuclei have the same number of protons, but a different number of neutrons. And I also want to look at hydrogen atoms. So let's start with the first interaction, or the first radiation, that's X-rays. So the principal contribution to scattering here is due to the interaction of X-rays with the electron density distribution. And in macromolecular crystallography, we typically assume that this electron density is spherically symmetric with some radial dependence. And that leads to scattering factors that look like in the next figure here. So here I plotted the scattering factor as a function of sinus theta over lambda. I also have the direct space resolution in some of the plots on top if you prefer looking at these values. And I plotted this here for three different types. So for oxygen, it's the red curve. For hydrogen, it's this brown curve. And for a negatively charged oxygen, so kind of an oxygen ion, it's the green curve. And so there are already several things we can observe here. The first thing is that these values are all positive. It's positive for the oxygen, it's positive for the ion. The second thing is that the value at scattering angle 0, so that would be here, corresponds to the atomic number, so or to the number of electrons. So for hydrogen, it's 1, for oxygen, it's 8, and for the negatively charged oxygen, it's 9. The second thing is, yet another thing is that we can see is that there is a decay. It looks a little bit like sum of Gaussians, this kind of curve. So there's a decay with increasing scattering angle. And the other thing is that for the ion, the scattering factors are different at small scattering angles. But if we go to larger scattering angles, there is a superposition with the values for the neutral atom. So we can also look at the scattering factors for more elements. So here in this plot, I have the scattering factor as a function of the element at 0 scattering angle. So it basically corresponds to the atomic number. And unsurprisingly, this will increase for the elements. And also I want to point out for the different isotopes, the scattering factor is identical because it doesn't care about the number of neutrons in the nucleus. So if we look at now at our table, so for neutral atoms, we saw that the scattering factor is always positive. For ions, it is positive as well, and the scattering factor at 0 scattering angle corresponds to the number of electrons. There's no difference for isotopes, and the scattering factor for hydrogen is very small compared to other elements. So what does it mean for maps? So that means that we have positive peaks for atoms, and hydrogen atoms are only visible at high resolution. So let's go now to the next type of interaction, which are neutrons. So the principal contribution to scattering here is due to the interaction between neutrons and the nuclei. So the nuclei, we can consider them as point scatterer. So as a consequence, we don't actually have a scattering factor as a function of scattering angle. We can forget about all this. What we have here is a constant, which is called B, not F, and it's called the neutron scattering length, not the scattering factor. And we can again plot this. Again, if we plot this as a function of sine theta over lambda, of course, we only obtain a flat curve. So here I have the scattering length for oxygen and hydrogen. So I did not plot it for the oxygen ion because it will be the same, because neutrons, they don't care about the charge of an atom because they interact with the nuclei. So it will be exactly the same location. Now, the interesting thing here is that the scattering length of hydrogen is negative. So that's different compared to X-ray. In X-ray, we only had positive values for the scattering factors. Now the scattering length can be positive and negative. And so let's again look for more elements. So here's the scattering length as a function of element. And we can see that there's almost a random distribution of the scattering length. And we have negative values, we have positive values. So for H, hydrogen, it's negative. For deuterium, it is positive. Then for carbon, nitrogen, oxygen, other common elements in macromolecules, we have positive values. And I think it's also interesting to be aware that for sulfur, the value is actually smaller than for carbon, nitrogen, oxygen. So sulfur will actually be a weak scatterer for neutron diffraction. So if we look at our table now, what do we have for neutral atoms? We have an almost random distribution of the scattering length with the atomic number, can be positive and negative. Ions is exactly the same as neutral atoms. We can have a significant variation between isotopes. One example is hydrogen versus deuterium. And another important thing is that the scattering length of hydrogen is of the same order of magnitude as other elements. So hydrogen will be like a heavy atom, carbon, oxygen, nitrogen in X-ray. So what does it mean for maps? So the one is that we will have positive and negative peaks in nuclear density maps. The second thing is that the relative peak height is different than what we are used to from X-ray crystallography. So as I said, hydrogen will have significant peaks, and sulfur, for example, is a weaker scatterer. And hydrogen will have negative peaks, deuterium positive peaks, so there might be cancellation effects depending on how the sample is constituted. So now let's move to the last type of interaction, that's electrons. And here the principal contribution to scattering is due to the interaction of the electrons with the electrostatic potential of the sample. So we have a positive potential from the nucleus and a negative potential from the electron density distribution. So the scattering factors of electrons are basically kind of related to those from X-ray. And we can actually derive the scattering factors for electrons from the X-ray scattering factors. And the important thing here is that we have a charge correction term for ions, and that looks like this. So the electron scattering factor for an ion is the electron scattering factor of a neutral atom plus a so-called charge correction term. So it's a bunch of constants times the charge over S square. And we see in a moment what effect that will have. So let's see first the scattering factor for oxygen. It looks quite similar to X-ray. Of course, the values itself are a bit different, but the overall behavior is the same. And what happens now for the oxygen, charged oxygen species? So here what will happen is we have, let's say we have -1 charge on the oxygen. So we have a constant. This is a positive value, -1 for the charge. And here we have s squared. So what happens is if we have very low scattering angles, this value here will tend towards -infinity. And that's what's happening here. So the scattering factor for a negatively charged oxygen will follow this orange curve. And so this is even more complicated than for neutron case, because now the scattering factor can have both positive and negative values. So part of it is negative. Here it will go up to the positive values. And so that means it can, depending on the resolution range of the data, this will affect the maps. So let's fill out our little table. For neutral atoms, the scattering factor is always positive. For ions, there's a singularity at a 0 scattering angle that can be positive or negative. For isotopes, there's no difference in electron scattering. And for hydrogen, the scattering factor is small compared to other elements. So what does it mean for maps? Neutral atoms will have positive peaks, and ions might have some cancellation. So I'm saying possibly cancellation because, again, this depends on the resolution range. So imagine you calculate your map, or you have experimental data that only include resolution up to, let's say here, which is maybe around 6 angstrom. So if you don't have any lower resolution, you don't have any contribution from this negative part of the scattering factor.
5 minutes remaining.
Positive. Okay, you will have a positive peak. And hydrogen, again, are only visible at high resolution. So knowing all this, we can now determine which of these maps corresponds to which experiment. So I want to give you maybe a minute to think about it, and maybe you can put some suggestions in the chat, or I guess the chat is not the fastest if you want. So for example, A is neutron, B is X-ray, C is electron. So I invite you to give it a try and try to figure out which of these maps corresponds to which experiment. So maybe while you think about it, I can talk a bit more about how I generated these maps. These are model calculated maps. These are not experiment, so no data were used. These are purely determined from a model. And so I used B factors of 20 for each atom. I used a negative charge on these 2 oxygen atoms at the carboxylate group of the aspartic acid side chain, and the unit cell was P1. I used a buffer around 5 angstrom over each from each atom. So I don't know if people actually post anything in the chat. Okay, we have one suggestion in the Q&A. So Jan says we have electron for A, B is neutron, C is X-ray. Okay, that's almost correct. So A, okay, electron, B, X-ray, C, neutron. Right, so that's the correct one. And let's see why that is the case. So A is the electron map, the electrostatic potential map. And I told you that I used as a model here a charged carboxylate. And so because this is charged, the density, there will be this partial cancellation of the density around the charged species. So that is telling us why this is the electron map. Okay, and then the second one is the X-ray map. So here we have density around basically every atom. And the last one is the neutron map. So you can see that the neutron and X-ray map are pretty similar, but there's one difference, and that's this CH2 group here. So you can see that the CH2 is not covered by, or the carbon atom even, is not covered by any density at all. And so this is due to the fact of the cancellation. So there's a CHH, and the sum of the scattering lengths for these for this group is almost 0. And so that's why this density almost vanishes. But otherwise, I agree that actually the X-ray and neutron map look pretty similar. And apart from this hint, it's difficult to actually
just 2 minutes left, please.
Okay, this is one last slide. So I hope I could explain the subtle differences between the maps. And now I want to tell you that, well, all these differences maybe are not even observable. So why is that the case? So map features also depend on a variety of factors, such as the B factors, so how the atoms move, static disorder, radiation damage in the case of X-ray and electron, data quality, and completeness. And so to illustrate this, I did a little one more calculation of a map. So again, here on the left, I have the electron, the map for the electrostatic potential, using half a negative charge on each of these two oxygen atoms. And what happens actually if I calculate the electrostatic potential map for the same group, which is neutral? So here the oxygen atoms are neutral, but I apply a B factor gradient. So instead of having a B factor of 20 angstrom square for each atom, I have 20 here at the C alpha, then I have 25 at the C beta, 30 at the C gamma, and then 35 at the oxygens. So completely, so I apply here a B factor gradient, which is a bit arbitrary, of course, but it represents a bit the fact that the side chains are often moving more than the main chain, and that there's a gradient of the motion. And so what will happen is, again, the density on the oxygen atoms will retreat a little bit. And the effect is almost the same as if I have a charged residue. So that means if you see disappearing density on the aspartic acid side chain, it does not necessarily mean, oh, this is a charged residue in electrostatic potential map. It could be also thermal motion. So this is all I wanted to tell you. I'm work in the Phoenix group, which is a collaboration of different laboratories. And apart from the main developers, there are, of course, many more people who are contributing to Phoenix. So again, I want to thank the organizers for giving me the opportunity to speak here, and I thank you for your attention.
Okay, fantastic. That was a really interesting tour through some of the mysteries of maps from all these different methods. I think we have time for maybe just one question from Slack. So Sony, do you have a question?
Yeah, so yeah, I think in the Slack it's just, David Briggs thanks you for the extremely clear and comprehensive overview of different diffraction data. And we have a question in Zoom, which is by Peter Moody, and it says, for electrons, how does the hydrogen scatter vary with resolution? What would hydronium look like?
Oh yeah, that's a good question. I actually did not plot the hydrogen. I think for hydrogen, the neutral hydrogen, it's very similar to X-ray, but it's actually a good question how that would look like for charged species, or even for any hydrogen atom that is covalently bound to another atom, because basically the charge will be deformed. It will be basically be a dipole. So I cannot answer this question, but it's very interesting, and to look more into that.
Wonderful. So thank you very much. And we'll move on now to our next speaker.